All right. So welcome to today's lecture. Today for chapter 10, we're going to talk about an introduction to Mendelian genetics. And Mendelian genetics gets its name from Gregor Mendel, who I'll talk about here in a little more detail. But Mendelian genetics gets its name from Gregor Mendel, who has the nickname the father of genetics. What a lot of people don't realize about Gregor Mendel is that he was a contemporary of Charles Darwin. That means that they lived at the same time. However, Charles Darwin, who's also known as the father of evolution, they never actually met. And this was, of course, in way before the days of the internet, and they never actually heard of one another. They never heard of each other. And another interesting thing is they were never acquainted with each other's work. And that's pretty astounding considering that they kind of solved some puzzles about inheritance and how things are passed on. And they both made some profound contributions to the area of science, the area of biology. I mean, their contributions are so profound that they get these titles, the father of genetics and the father of evolution, but they never met, they never heard of each other, and they never even were aware of the other's work. And so I cannot imagine what kind of, you know, mind-blowing summit it would have been if Gregor Mendel and Charles Darwin could have met and had, like a week just to talk to each other about all the things that they were contemplating, much less actually discovered, just the things that they were thinking about. It would have been probably one of the greatest summits in all of science, this exchange of ideas between Charles Darwin and uh, Gregor Mendel. But unfortunately, it never happened. So we can only imagine what that would have been like. Well, let me talk a little bit about Gregor Mendel, because I am going to talk about Charles Darwin, not in this lecture, but I'm going to talk about him in a future lecture. Let me talk a little bit about Gregor Mendel. There is a picture of him you can look at if you're following the slideshow, slide number 3, or on page 168 of your textbook. So I'm going to go to slide number 3 right now, show you a picture of Charles Darwin. This is a very famous picture of him, and you may notice he's dressed in clergy garb. He was a clergyman, he was a monk. He was a monk, and he was sent to study at the University of Vienna. He was sent there to study science and mathematics. He wanted to become a scientist and a teacher. Unfortunately, Gregor Mendel did not succeed in completing his university education, not in the way he had hoped. There may be some evidence that Gregor Mendel suffered from extreme test anxiety, or he may have had a learning disability, because even though he was a good student and he studied hard and he worked hard, he never successfully passed his exams. He had a hard time passing the exams. So if you need somebody to inspire you, think of Gregor Mendel. He had a hard time with school as well, although he still persevered. While he was in the University of Vienna, he began to take an interest in plants. And his interest in plants served him very well later, especially as it came along with his interest in science, because his interest in plants, along with his interest in science, eventually led to this title that he gained as the father of genetics. We know that Gregor Mendel was especially involved with garden pea plants. He was especially involved with garden pea plants. And it turns out that pea plants are perfectly suited to study heredity. And heredity is the science of how things are passed on. One of the reasons that pea plants are so well suited for the study of heredity is because many of the varieties of plants, of pea plants, they have very distinguishable traits. So like they might have something like purple plants or purple flowers or white flowers. They might be tall flowers or they might be or tall plants or they might be short plants. These are things that we can very easily observe and we can count very quickly, how many purple flowered plants do you have? How many tall plants do you have? Things that we can distinguish very easily and that we can count easily. Pea plants are also relatively small. They don't take up a lot of space. They're easy to grow and they produce a lot of offspring very quickly. So, why this is beneficial is that you can go through many generations in a relatively short amount of time. So, if you're studying heredity, the science of how things are passed on, then these traits are very useful for that reason. Another good thing about pea plants is the way that they are put together. It's easy to manipulate their reproductive organs so that they can pollinate or fertilize the way you want them to fertilize. They can also self fertilize. So, these are some reasons why pea plants are a good model to study heredity, and they're probably the very same reasons that Gregor Mendel chose them in order as his model organism to study heredity. We'll come back to this slide, a lot of information on this slide, but let me show you a little bit let me show you a picture of a pea plant to tell you why this is such a good model or why it was such a good plant to study. And I'm going to take this picture directly out of your book. I don't have a slide of it, unfortunately. But here is a picture of a pea plant directly out of your book. So if I look at this diagram of the pea plant, this is a kind of like a pea plant cut in half. And here are the petals. And here in the middle are the reproductive organs of the pea plant. I'm going to draw them a little bit closer on my paper so you can kind of see what we're looking at. But if I take a pea plant, and here's the petal, oh, I misspelled that, P-E-T-A-L. I spelled it petal like a bicycle pedal, P-E-T-A-L. Okay. When you look at a pea plant, you have the girl parts, which is like a long tube, and that girl part, those are called the carpels. Then on the side of the female parts, you have these long filaments, and at the end of the filaments, you have these little fuzzy things called anthers. This is the source of pollen. And in case you didn't know, pollen is plant sperm. So the anthers are the male organs, and the carpels are the female organs. Now, the way that you can manipulate this flower is you can allow it to self fertilize. So the pollen can literally dust on top of the female carpel, or you can cut it and you can remove the male parts and then you can fertilize it manually. So the design of the pea plant was such that it would be relatively easy to either allow it to self fertilize or cut and remove and do manual fertilization. So when Gregor Mendel was looking for something to study heredity with, pea plants fit all of what he needed absolutely perfectly. Now, the story of how he kind of studied his pea plants, what he ended up doing, and my diagram is really small. I'm going to see if I can make it bigger. Yeah, I can make it a little bit bigger. Okay. How Gregor Mendel started out his experiments was he ensured that he started with what we call purebred or homozygous varieties. So he started with what were purebred or homozygous plants. And so what he did was he took plants with distinguishing characteristics. In this case, he picked plants that had either white flowers or purple flowers, and he allowed them to self-fertilize over several generations. So by allowing plants to self-fertilize for several generations, he ensured that he was working with purebred plants. So he acquired them via self-fertilization. He called those his parent generation. So he had a plant with white flowers. Oops. And he crossed it with a plant with purple flowers. Now, mind you, these were purebred. So he took his purebred purples and his purebred whites and he crossed them in order to see what he would result with. So he called this his parent generation. Or sometimes we call it the P1 or the parent generation. The result of this was 100% of the flowers of the offspring had purple flowers. He referred to this first offspring as the F1 generation, or the first filial generation. Filios is Latin for offspring. So he called this the F1 generation. Then he allowed this generation to self-fertilize. He allowed that generation to self-fertilize, and he was really surprised at what he received. The second filial generation, or the F2 generation, he received approximately 75% purple flowers and approximately 25% white flowers. So his offspring ended up looking like the grandparents in roughly a 3-to-1 ratio. When he allowed this generation to fertilize, he also got a mixture of purple and white flowers in a different type of a ratio. His observations over many generations led him to develop what is now referred to as Mendel's postulates. So his observations over many generations led to what we call Mendel's postulates. Mendel's postulates are somewhat stated on page 170 of your textbook. If you're following in the slideshow, this is slide 13 through 16. So if you're following the slideshow, it's slide 13 through 16. But I'll go through Mendel's postulates for you, and I'm going to state them just a little bit differently. But they'll have the same meaning as what's in your textbook. So here we go. What were Mendel's postulates? Gregor Mendel said that every trait requires two factors. Every trait requires two factors. One factor is donated by parent number 1. The other factor is donated by parent number 2. So every trait requires two factors, one donated by parent number 1, the other donated by parent number 2. The second thing he said is, during gamete formation, those factors segregate so that each gamete only inherits one factor. So during gamete formation, factors segregate so that each gamete inherits only one factor. Dominant factors, I'm getting ahead of myself. Fertilization is the event that restores paired factors. Fertilization is the event that restores paired factors. Dominant factors completely mask recessive factors. So here's kind of a restating of Mendel's postulates, the ones that are in your book and the ones that are in the slideshow. This is a little bit of a restating. When Gregor first proposed these 4 statements, people were not sure how to receive them. You have to understand, or at least appreciate, that at the time that Gregor proposed these statements or these postulates, this is a postulate is like a hypothesis. At the time that Gregor proposed these ideas, people did not know about DNA. Believe it or not, you know a lot more about DNA than what Gregor Mendel ever knew about DNA. He had never heard of DNA. He didn't know anything about chromosomes. He didn't know anything about adenine and guanine. He had no idea. He just knew that somehow, based on his observations, that heredity was controlled by some indescribable factor. That's the best that he could use to describe it at that time without any knowledge of DNA. And so when we look at these statements, it's really kind of mind blowing. We really have to appreciate how much he was able to describe without ever using the word DNA in any part of it. When we look at this very first statement, we can change one word and modernize this entire statement. Instead of saying that every trait requires two factors, we can say every trait requires two genes. One donated by one parent, the other donated by the other parent. So everywhere you see the word factor, you can write the word genes. During gamete formation, genes segregate so that each gamete only inherits one gene. We saw this in anaphase 1 of meiosis. During anaphase 1 of meiosis, this is what we refer to as the law of segregation. The idea that factors separate so that each gamete only inherits one copy. It's also where we described you go from diploid to haploid. Fertilization is the event that restores paired factors. So when you have a sperm and an egg to form a zygote, well, the parent number 1 carries 23, if we're talking about humans. Parent number 2 carries 23, so that the zygote inherits 46 total chromosomes. In other words, we have haploid is what I mean to say. Haploid plus haploid creates the diploid, restores the chromosome number. Dominant factors completely mask recessive factors. This is one we haven't talked about yet, and this was observed in the F1 generation of his pea plants. What we understand now is that genes have alternate forms that we call alleles. So genes come in alternate forms that we call alleles. Like, for example, you might have a gene for flower color. The options for flower color were purple, which I misspelled, purple, or white. So instead of saying a purple gene or a white gene, we say that the gene is for the trait of flower color. The forms that the flower comes in is purple or white. So you have the purple allele or the white allele. You might have a gene for plant height. Okay, that's the trait. But your options is you can be tall or you can be dwarf. So what you inherit, we say, is the tall allele or the dwarf allele. What Gregor Mendel observed is that some alleles have the ability to mask or hide other alleles. So the ones that have the ability to cover up or hide, he called them dominant. Dominance doesn't have anything to do with like aggression or being a desirable trait or even being the most common trait. It just simply means that you can cover up something else. Recessive traits are not necessarily weak, they're not bad, they're not less common. It just simply means that if a dominant one is present, then they can be covered up. That was Mendel's the words that he chose in order to explain what he was observing. So what we know from Mendel's studies is that purple plants were dominant, which we use a capital P to represent, whereas white plants or white flowers were recessive. We keep the same letter, we just use capital and lowercase. So like for tall, we might use T for tall and little t for short. Dominant factors are indicated with a capital letter. Recessive factors, same letter, it's just that they're lowercase. So like I mentioned, a big P and a lowercase p. Now, of the traits that Gregor Mendel observed, there's a table on page 168, and I don't have it on my slideshow, unfortunately, but I do have the table on page 168. It's table 10.1 to show you all the observed traits that Gregor classified as dominant versus recessive. So I'm going to show this to you from my book. This is page 168. And I've written in my book here. So here was the gene for flower color. The trait or the gene for flower color came in two different alleles, either purple or white. It turned out that the purple was the dominant form and the white was the recessive form. Another gene or another trait that he observed was the color of the seeds. They could be yellow or they could be green. He also observed the shape of the seed, whether it was round or whether it was wrinkled. And again, this chart tells you which was the dominant, which was the recessive. He looked at the color of the pods that the peas grew in. He looked at the shape of the pods, even the position of the flowers. Were they along the stem or were they only at the ends? And finally, he looked at the height of the plants. With these 7 traits, over many, many, many generations, Gregor Mendel was able to figure out basically the foundation of what we call completely dominant genetics or Mendelian genetics. It was later on when a scientist by the last name of Punnett developed a simplified way to explain Gregor's crosses and the results of those crosses. Based on his method, now we have a diagram that we can fill in called a Punnett square. And you've probably seen these before if you've taken a high school biology class. So Punnett squares begin on page 171 of your textbook. Or if you're following the slideshow, I'm on slide number 27. So we'll use a Punnett square to explain Mendel's observations. So Mendel crossed two purebred pea plants. One had purple flowers, one had white flowers. So we're going to represent the purple allele with a capital P. And we're going to represent the white one with a lowercase p. And we're going to take into account Mendel's law that says for any trait, you need two alleles. You need two factors. So parent number one with purple flowers would have had two capital P's because he was a purebred. Another word for this is homozygous dominant in this case, because he has two dominant alleles. Parent number two had white flowers, so they would have had two lowercase p's. They were also purebred. So we can still describe them as being homozygous. But because they have the lowercase alleles, they are homozygous recessive. So just as a reminder, purple is dominant, white is recessive. So the method of using a Punnett square is first thing that we do is we indicate a cross between two parents. And a cross is a mating event. A cross is a mating event. And we indicate that by putting a little X between what's called the genotype of the parents. So when you represent their alleles, that's called the genotype. What they look like, so this is purple. What they look like is called their phenotype, like a physical characteristic. So the phenotype of this one would be white flowers. But their genotype are the actual letters that we're using to represent them. So when you do a Punnett square, you indicate the genotype of the parents. And then you imagine that the parents each go through meiosis. Now remember, in meiosis, you make 4 total gametes. Half of them are going to have this information, half of them are going to have that information. So here is, if this is the dad, that's sperm number 1, sperm number 2, sperm number 3, sperm number 4. Egg number 1, egg number 2, egg number 3, egg number 4. It just so happens that of these 2 eggs, or these 2 sperm, there's duplicates. Of these 2, you have a duplicate. And of those 2, you have a duplicate. So we can cross out the duplicates. And that's going to make our Punnett square pretty simple. We'll have one possible gamete, bring down your X, and another possible gamete. So our Punnett square is going to have 1 by 1 square. I put the sperm on the outside over here, the egg on the outside over there, and the interior of the box represents one possible mating event. So if this sperm fertilizes that egg, that is the resulting individual. What they look like is that they have purple flowers. But they are what we call heterozygous. because they have one capital letter and one lowercase letter. In other words, this one masks the white flowers. So you don't have purple and white. These purple flowers look exactly like the purebred purple flowers, which is what we saw in Gregor Mendel. This is the only outcome. So in his F1 generation, we would have seen 100% of those purple flowers. We'll use a Punnett square to explain what we saw when we crossed the F1 generation with the F1 generation. There's a lot of detail on this handout, on this sheet of notes. Type a number 1 for me when I can go on to the next page. Okay, good. Good, you're keeping up, good. So the F1 generation, the result, page 5. The result of the F1 generation, they were all capital P, lowercase p. That was their genotype. And they all looked as purple flowers. When Mendel crossed 2 of the F1 generations, so he did an F1 by an F1 cross, his result was roughly 3-1, if you remember that from our earlier notes. Well, how does that work? Well, again, we take our capital P, lowercase p, times capital times lowercase. Half of the gametes are going to look like this. Half of the gametes are going to look like that. Same thing over here, half of your gametes, if I can cover it up, half of your gametes are going to have this information. Half of your gametes are going to have that information. So I've got a 2 by 2 square with a total of 4 squares. So 2 up and down, 2 going across, with a total of 4 squares. Now on the outside of my squares, I put my possible gametes. And I would recommend that you make sure you can tell your capital letters from your lowercase letters. Don't get sloppy on me. Make sure you can tell your capital from your lowercase letters. And so this square is a little more complicated than our last square. We have 4 squares on the outside. So the way we're going to fill this in is if this P fertilizes that P, this is your resulting individual. And remember, these ones over here are sperm. These are eggs on top. If this sperm fertilizes that egg, by convention, we always write the capital letter first. There's your resulting individual. This sperm, if it fertilizes that egg, there's your resulting individual. This sperm with that egg, there's your resulting individual. So when we looked at them, this one is purple because you have 2 capital P's. This one is purple because remember the capital P can mask the recessive, so he's purple. Same thing here, we've got a masking going on, so he's purple. But the only information that this one has is for white flowers. So we ended up with approximately 75% purple, 25% white. That was what we call our phenotypic ratio. Is 3-1. But our genotypic ratio is 1 to 2 to 1. In other words, we had 3 that looked the same, but their DNA, their combination, there was one that was completely dominant. There are 2 that are heterozygous, and there is one that is completely recessive. So it's interesting that our DNA is a different ratio, but how they appear looks like this. I'm going to stop here for today because I have a short video to show you. It's about 10 minutes long that talks about Gregor Mendel in a little bit more detail. And if you're having trouble filling in the boxes, there's an animation that also talks about how you fill in the boxes. But before I switch to the video, do you have any questions for me?
No, ma'am.
None from Brandon. Brandon, I'm glad you're back.
I have no idea what's going on with my Wi-Fi this morning.
Is it acting crazy?
I couldn't hear, and then like even my screen froze for a little bit. But it finally, I just had to restart the router.
Oh, yeah, that's a good idea too. Well, I'm recording today, so if you missed the front part, you can always go back and rewatch it.
Okay, yes, ma'am.
But I'm glad you did not give up. Good job persevering.
Yes, ma'am.
Okay, well, I'm going to show the video. And I'll post the link to this video also in Blackboard, so if you want to watch it again later, you can. So let me know right now, do you see the video on your screen? Yes or no?
No, we see you.
You still see me?
Yeah.
Did I not do this right? Okay. I don't even see me. Okay. Okay, how about now?
Yes, we see the video.
Thank you, Nancy. Otherwise, I was afraid I was going to have to act out the video. And my acting skills are not good. All right, here's the video. It's about 15 minutes in length.
Hi, it's Mr. Anderson, and welcome to Biology Essentials video 29. This is on Mendelian genetics, and it's called Mendelian genetics. It's named after Gregor Mendel. In biology, there are 2 famous names: Darwin, who was famous and controversial in his own time, and Gregor Mendel, who died in obscurity. But both of them made huge advances in the field of biology. And Gregor Mendel in the area of genetics. If they could have gotten together, those 2 theories, when they finally came together as modern synthesis, it was really powerful. Unfortunately, he dies in obscurity, but this is what he did. He was crossing pea plants, and so he can take one pea plant and you use a paintbrush to transfer pollen from one to another so you know who are the parents. He would then create offspring as a result of that. Now, peas are great because they have a number of different characteristics, and more importantly, you can make a lot of them really quickly. So in a pod, each of these peas is an actual new organism. So you can plant those and see how they grow. And so he figured out a lot of genetics as a result of that. And so genetics in this podcast, I'll talk about are Mendelian or simple genetics. He identified the gene and he came up with 2 laws, the law of segregation and independent assortment. I'll talk about those. We'll also have some problems. So I'll have some practice problems that you can try and I'll show you how to work those out. And we'll finish with genetic disorders. The example I'll talk about is Huntington's disease. And with genetic testing, it opens up this whole idea of ethics and privacy. Now, what won't I be talking about? I won't be talking about linked genes. In other words, if genes are ever on the same chromosome or on a sex chromosome or caused by multi-genes, it's really complex. And so I'll talk about those in the next podcast on advanced genetics. But know this, that simple Mendelian genetics, the rules are simple rules of math, rules of probability. And if you understand those and how to do the first cross that I show you, you should do well in most any of the crosses you get in genetics. And so this is what Mendel did. He crossed purple flowers with white flowers, and he got purple flowers. Now, a few things that you should know. The first cross in any genetic cross is called the P cross, or the parental cross. And then the offspring of that are called the F1, or the filial one, or the offspring of that cross. And so at the time of Mendel, everybody believed in this idea of blending, that if you cross two parents, so mom and dad, their kids look a lot like them. And so it's maybe a blending of they didn't know that they were genes, but something inside of them. And so when he crossed purple with white and got purple flowers, that made total sense back then. This kind of fits in this blending. But what he did next was he crossed these purple flowers with themselves. And what he got was a 3-to-1 ratio of purple to white. And that white returned just as crystal white as that first white was to begin with. And so what he said is that there's a character, a trait that's passed through here, but we now identify that as a gene that's carried distinctly through each of those generations and then shows up again. Now, if you know anything about genetics, you'd know that in simple Mendelian genetics, it looks like purple is dominant, white is recessive, but we know that because of the work of Mendel. And so this is what the second cross is, the one that was puzzling, and this is what he eventually figured out. So these offspring right here were hybrid for the trait. And so if we look at the parents would be big P, that represents one purple flower. Little p represents the white flower. And so each of these would be big P, little p. And so you can use a Punnett square. A Punnett square, you put the pollen or the male here, the female right here, and each of these genes get a certain column. And so this would be one parent, big P, little p. And so you simply write that across. So we got big P and little p here. We got big P and little p here. So this represents the two different parents crossing with each other. And then we simply figure out what comes. So here's big P, big P. So we get a big P from one, a big P from the other. So that'd be big P, big P. Since big P is dominant, we get a purple flower. On the next one, I'm going to take a big P from here, a little p from here. And so that would be still purple because this one is a dominant allele or a dominant gene. Down here, we get a big P from here and a little p from here. And so that's purple. And the reason we get white flowers is that you get a little p from both of the parents. And so the neat thing about a Punnett square, and that's what this is it not only allows you to quickly do the probability, but it shows you the percent we should get in those offspring. In other words, 3 of them should be purple and 1 of them should be white. So we should have a 3-to-1 ratio. And if you ever get a cross like this in a problem, for example, a cross like this, just if you're not sure what the offspring are going to be, just do a simple Punnett square where these two alleles or versions of the gene are going to be on the top and these two are going to be on the side. And we'll do some practice problems in just a second. And so what are Mendel's laws? Well, the two things that he figured out are the law of one, Mendel's law of one, it's called the law of segregation, and Mendel's law 2 is the law of independent assortment. And so let's start with law of one. And so if we ever, and I got a coin here because the actual way you get genes are almost like a coin flip. So if you think about a coin, it has heads on one side and tails on the other. When you flip a coin, what are the odds that you're going to get heads or tails? Well, it's a 1 in 2 probability that you'll get heads. Same thing with genes. And so if this is that F1 generation, and this shouldn't be B, this should be P. So we'd say this is big P, little p. What are the odds that the offspring are going to get a big P? Well, it's a 1 in 2. What are the odds that they're going to get a little p? It's 1 in 2. And so that separation of those two alleles is called segregation. And so this idea of segregation says that there's a 50% chance you're going to get either of these genes. And so that's segregation. They separate, and it's just random chance. The next one is the law of independent assortment. The law of independent assortment says that this gene, the gene that causes, for example, hitchhiker's thumb, which is where your thumb actually bends back, and the gene that causes an attached earlobe, so right here, I've got a free earlobe, those two traits don't affect each other. In other words, they sort independently. And so we can work problems without mixing these two together. They're going to not influence one another. Now, sometimes we'll find, for example, that some things do travel together. So you'll notice that people who have red hair also have freckles. And that's because those two genes are actually found on the same chromosome, and so they seem to travel together. And so we're not going to deal with linked genes again. We'll do that later. And so independent assortment means that traits don't affect each other. And so what I'm going to do next is I'm going to leave this for a second. These are 6 problems that I'll work through. But if you want to work these, you could pause the video at this point, and then you could come back and start the video again and see me work through each of these. So I'll pause. All right, so let me go through these. Question 1, a coin is flipped 4 times, comes up heads each time. What is the probability that the next coin flip will come up heads? Well, everything that's happened in the past can't influence anything that's going to come in the future. And so it's a 1/2 probability that you'll get heads. In other words, you could have 10 kids, they could all be boys. What are the odds that the next one's going to be a girl? It's still a 1 in 2 probability. Let's look at the next one. And so we've got some things up here. Round peas are going to be big R, and wrinkled peas are going to be little r. Yellow will be big Y, and dominant is going to be, or green is going to be little y. Generally, whatever is the dominant trait, we give that the capital letter. In this case, round gets the big R, and yellow gets the big Y. And so question number 2, classify the following as heterozygous or homozygous. Heterozygous means you have different genes or different alleles. Homozygous means you have the same. And so this first one, big R, would be homozygous dominant. They are the same. This would be heterozygous, and we also sometimes refer to that as hybrid. The next one would be homozygous recessive, and the next one would be heterozygous yellow, homozygous for the round. So it's going to be heterozygous and then homozygous dominant on the next one. So that tells you the alleles that you have. Let's look at number 3. What's the phenotype of the following? Well, this right here is going to be the genotype. In other words, big Y, little y is going to be the genes that you have. What's going to be the phenotype? Well, that's physically what you look like. And so for the first one, this one right here, even though its genotype is big Y, little y, or it's heterozygous for that, its phenotype would be yellow. So this is going to be yellow. This one's going to be round. This one's going to be green. And this one here is going to be yellow round. Phenotype is physically what you look like. Let's look at the next one, number 4. What's the probability of this cross? So we have 2 round seeds producing wrinkled seeds. Well, like I said before, if you ever get one of these, it's a simple monohybrid cross. I would always do a Punnett square. And so we put big R and little r on one side of my Punnett square. Big R, little r on the other side of my Punnett square. So what are the odds that I'm going to get wrinkled seeds? Well, there's a little r here, a little r here. And so there would be a 1 in 4 probability that we'd have wrinkled seeds, because this one's going to be round, this one's going to be round, this one's going to be round as well. And so again, if you ever get a simple cross like that, do a Punnett square. Let's look at the next one. What's the probability that this cross would produce green seeds? Well, I do the same thing again. Big Y, little y, crossed with little y, and we're looking for green seeds. Green seeds, remember, are going to be little y. And so I could get it here, I could get it here, and so that is a 2 in 4 or a 1 in 2 probability that we're going to get green seeds from that. And so even though you might think you're super smart, do a Punnett square. You're never going to miss the problem then. Now, this is a problem that we'll sometimes get on the AP Bio test as well. If these parents are crossed together, what are the odds that you'd get that? Well, to do this one, you'd have to actually set up a pretty intense Punnett square. And so if you get one like this, don't do a 4x4 Punnett square. Just work each of them individually. And so what do I mean by that? Well, let's start on the R's. What are the odds that if you produce this and that, you could get that? So let's do an R's first. So big R, little r, crossed with big R, big R. What are the odds that we're going to get big R, little r? Well, neither of these, but this is a big R, little r, this is a big R, little r. And so the odds of these 2 parents producing these offspring is going to be a 1 in 2 probability. So I'm going to write that right over here underneath the 1 in 2 probability. Now let's work the Y's. So let me get a different color. So if we do the Y's, what are the odds that these 2 parents are going to produce that offspring? Well, let's do those together. So here are the parents, big Y, little y, crossed with big Y, little y. So this is going to be big Y, little y, little y, big Y, little y, big Y, little y. So what are the odds that we're going to produce again? Big Y, little y. Well, it's a 2 in 4 or a 1 in 2 probability. So I'm going to write 1 and 2 here. Okay, so now instead of doing this huge unwieldy 4x4 Punnett square, what I've done is I've almost got there. Because the odds of getting this are 1 and 2, the odds of getting this are 1 and 2. So what are the odds of getting both of those? I simply multiply those together. And it's a 1 in 4. In other words, what are the odds of rolling one or flipping the coin and getting heads? 1/2. What are the odds of flipping 2 heads in a row? It's a half times a half or a fourth. And so you can solve problems like this just using what's called the law of multiplication. These two things have to happen. So I hope you did well on those problems. Last thing I want to talk about is disease. And a nasty disease is called Huntington's disease. It's named after the person who identified it in the 1800s. But essentially what you get is degeneration of the nerve fibers in this portion of your brain. And so what happens is eventually you start to get uncontrollable shakes. You can't really walk. Eventually you die as a result of that. Now, the problem with Huntington's disease is you don't know you have it until you're middle-aged. So I could have Huntington's disease right now. I'm going to die as a result of this disease, but I don't know it. And so I've already had kids. I've already passed the genes on. A famous person who had Huntington's disease is this guy. His name is Woody Guthrie. You probably know him. He wrote the song, "This Land Is Your Land. This Land Is My Land." And he died as a result of having Huntington's disease. Now, it's a dominant trait. In other words, if you are this, you get Huntington's disease. If you are this, you don't. And so let's look at a pedigree. A pedigree shows you how a disease can be passed down through organisms. And so on a pedigree, a square is always going to be a male, a circle is going to be a female. And if you ever have a horizontal line between them, it means that they had offspring. And so this is the grandparents in this case, and they had a boy, and then the next kid they had was a boy, and then they had a girl, and then they had another girl. And you can see that this girl, for example, had her own family. But you can trace the disease through it. In other words, since this parent right here is big, let's use a different color, big H, little h, and this one is little h, this big H was actually transferred to the son. It was not transferred to this son. It was not transferred to this daughter, but it was transferred to this daughter over here. And so the odds of passing it on are 1 in 2, and you can see that 1 in 2 of their kids had that. And if it's a dominant disease like this, lots of times we'll see it in generation after generation after generation, but you've reproduced already by that time. And so it's almost too late. Now, where does this become an ethics issue? Well, we now have a test for Huntington's disease. And so Woody Guthrie had a number of kids. One of those is named Arlo Guthrie, who's also a famous folk singer. And so Arlo Guthrie may have the Huntington's gene. He has a 1 in 2 probability of getting it. We now have a test that can figure out if you have that gene, but it'll influence your life in the future. And so would you want to know that you're going to get a disease that will cause a nasty death as a result of that? There's not a lot of treatment for Huntington's disease or not. And would your insurance company want to know that as well? And so again, the genetics behind simple Mendelian genetics are fairly simple, but it opens up all these moral issues. And I don't have an answer for any of those questions, but it's something we're going to have to deal with in the future. And so that's genetics, Mendelian genetics, and I hope that's helpful.
Good grammar.
All right, so that was a little bit of an expansion on what I had talked about today. He did some more complicated Punnett squares. We'll do some of those sample Punnett squares the next time I see you on Thursday. He also mentioned at the end something called Huntington's disease. If you are still looking for a topic for your oral presentation, Huntington's disease and the genetics of how it's passed on, what are some of the concerns about Huntington's disease, and what is it like to have Huntington's disease? This is a great topic for your oral presentation. Speaking of your oral presentations, while I have you the last few minutes for class, there is no Zoom meeting for lab this week. Instead, I'm giving you the time to do library research and prepare your oral presentation. So no lab meetings this week. Check your inbox in course messages in lab. You'll see that I mailed to you the oral presentation grade sheet. Also check the week 14 folder in lab. You'll see that there's a topic sign-up sheet. The only point of the topic sign-up sheet is so that two people don't talk about the same thing. The topic sign-up sheet doesn't mean that if you sign up first, you're going to speak first. It just means tell me what your topic is so that the other people in the lab section will know not to pick that topic. All right. Do you have any questions for me before we're done?
Yes, I do. Since I'm alone in the lab, do I still have to do that topic sheet, or can I topic anything?
No, you can do that. Now, Nancy, oh, I already asked this. I was going to say you can join us on Thursdays, but you work.
Yeah, I do.
Okay. So, yeah, no, you don't have to do a topic sign-up sheet.
Okay, thank you.
You're my only one. So,
Yeah.
me.
Okay, cool. Thank you.
Any other questions? If not, well, thank you for your attention today.
Thank you.
And I will see you on Thursday.
Okay, bye.
Bye-bye.
Okay, Miss Ware, I was gonna ask you something.
Yes.
Or there was the chapter 12 quiz I was due yesterday.
Did we do that together in class?
That's what I was gonna ask you. I was just I didn't do it because I was like, I'm positive that we did it because I downloaded the document and seen the questions and I was like, I'm pretty sure we done. I was just wanting to double check to make sure. Because whenever I clicked on the chapter 12 quiz, it gave me the option to start or to turn something in.
Yeah, no, you don't have to do it. You were already there that day. I'll update the grade book.
Okay, yes, ma'am. I just had a question about that.
Okay.
Okay, thank you.
Thank you.
Have a great rest of your week.
You too. Bye-bye.